Hello everyone, today we're going to talk about transcription and translation. In our body, we have tons of different types of proteins. All of these proteins in our body, they were made by two processes taking place inside of your cells. A process known as transcription and translation. Transcription takes place inside of the nucleus, while translation takes place outside of the nucleus in the cytoplasm of the cell. Transcription is the process of reading our DNA and transcribing it and making a messenger RNA. Once a messenger RNA is made, it's going to be released out of the nucleus via the nuclear pores, and it's going to bind with an organelle in the cytoplasm known as ribosome, where the messenger RNA is going to get translated into a protein. To simplify the idea of transcription and translation, we use this analogy right here to kind of explain the concept behind transcription and translation. Let's say that you want to bake some chocolate chip cookies. To begin baking those chocolate chip cookies, you have to know what the recipe entails. And so therefore, you go ahead and you find your cookbook. Now, the cookbook definitely has tons of different recipes. Are you going to read all the recipes just to make chocolate chip cookies? Of course not. You will look at the index, figure out what page is the chocolate chip recipe, and you're going to go ahead and start reading only the chocolate chip recipe. Let's say that recipe is on page 70 and 71. You're just going to read those two pages. Then you're going to start writing down the recipe on an index paper, on an index card. Now, you can go ahead and start making modifications to the recipe. Let's say the recipe is asking you to add white sugar. You don't want white sugar, so you replace it with brown sugar. So you can definitely make modifications to that recipe. After you've made your modifications, you go to the kitchen and you start bringing all the ingredients that you need, and you start combining those ingredients to bake your chocolate chip cookies, and that would be your final product. This is kind of what's happening inside of our cells. Our DNA is like the cookbook. So our DNA contains tons of different genetic information. Each gene could basically code for a specific protein or a specific trait. Now, let's say that we want to make our hemoglobin protein, which is the protein found in your red blood cells. To make that hemoglobin protein, are we going to read our whole entire DNA strand, which is 6 feet long? Of course not. We have to find exactly where the hemoglobin genetic sequence is at within our DNA. Let's say it's right here. Once you've located it, that's the only sequence you are going to read. You are going to read that sequence and you're going to make a messenger RNA. After you make the messenger RNA, which is kind of like the recipe that you wrote down, you can actually modify it. So you can modify the messenger RNA. You can erase some parts of the messenger RNA. You can add things to the messenger RNA. After you finish modifying the messenger RNA, it will leave the nucleus. So here is the nucleus. So the messenger RNA leaves the nucleus and goes to the cytoplasm. And it meets with the organelle known as the ribosome. Think of the ribosome as the kitchen. Now you are ready to assemble all the ingredients to make your hemoglobin protein. Proteins are made out of amino acids. And so what you're going to be doing to your messenger RNA is you're going to read that messenger RNA, and start bringing the amino acids that are needed, and you're going to start linking those amino acids. Once those amino acids are linked, then you will have your final product, which is, in this case, our hemoglobin protein. Our DNA is known to be a double helix, so we have two strands. Those two strands are not identical, they are complementary to one another. So if one strand reads A, the other strand is going to have a thymine base. So as you can see, they are complementary to each other. Now, one strand of our DNA is called the non-template strand, and one strand of our DNA is known as the template strand. As I said, to go through the process of transcription, we're going to have to read our DNA and we're going to have to transcribe it into our messenger RNA. Now, are both strands going to be read? And the answer is no. Only one strand of your DNA will be read to make the messenger RNA. Which strand is that? The template strand. So if this is the non-template, then ignore the non-template strand and only read the template strand. So if your template strand has a thymine, then your messenger RNA is going to have adenine. If your template strand has guanine, your messenger RNA is going to have cytosine. If your template has adenine, you're not going to have thymine on a messenger RNA, you're going to have a uracil. And so you're going to start building your messenger RNA by reading only the template DNA strand. The process of reading your template DNA strand and making a messenger RNA strand out of it is a process known as transcription. Transcription looks something like this. There are actually 3 steps that we have to go through to build that messenger RNA strand. We have to go through a step known as initiation, elongation, and termination. So 3 steps we have to go through. Here is the DNA strand with both the non-template and the template strand twisted around one another. As we said, this DNA strand contains tons of genetic information. Now, you do not want to read your whole entire DNA strand. You only want to read where you have the specific genetic sequence for that specific protein that you want to make. So you have to find a specific sequence on the DNA known as the promoter DNA. This is the sequence that you will have in front of the gene that you want to read. So this is the sequence that must be found. Which enzyme helps you find that starting sequence for transcription, which is the promoter? An enzyme known as RNA polymerase. RNA polymerase sticks to the DNA and it will stick to the promoter site so that transcription can go ahead and begin. Once RNA polymerase sticks to the promoter sequence on your DNA, as you can see, the DNA helix starts to separate. And as we said, we are only going to read the template strand. So ignore the non-template and only start reading the template strand. And you are going to start building your RNA. And that process is known as elongation. Why elongation? Because you're going to keep reading that template strand and building a longer and longer messenger RNA. And you can see right here, the messenger RNA is growing. And the RNA polymerase keeps sliding down your DNA. Now, Are we going to keep going through that step elongation forever? No. Anything that starts has to come to an end. And so once RNA polymerase hits a sequence on your DNA known as the terminator sequence, everything comes to a stop. RNA polymerase will step away, and you can see your DNA just goes back to looking normal, and we have our messenger RNA right there, and that will be the termination stop. So to begin transcription, RNA polymerase must bind to the starting sequence known as the promoter, and that promoter sequence is within your DNA. Once promoter binds to the RNA polymerase, transcription can begin, and you can start making and growing your messenger RNA. To stop transcription, you have to hit a sequence on the DNA known as terminator. After the DNA gets transcribed into a messenger RNA sequence, and again, all of this is happening inside of the nucleus, transcription happens inside of the nucleus, that messenger RNA, before it leaves the nucleus, it has to be processed. Remember how we said after you write down your recipe, you go ahead and you modify it? This is kind of what's happening right there. You have your recipe written down, which is your messenger RNA, and now you want to go ahead and modify it. So what's going to happen is there are sequences within the messenger RNA that gets removed, and those are known as introns. Those are not going to code for amino acids, and so we remove them. Whatever sequences are going to be left over are known as exons. Those exons are the ones that are going to exit the cell. We also add a cap and a tail. Those are a bunch of sequences that we add to the beginning and the end of our messenger RNA, and the cap and the tail helps protect the messenger RNA because that messenger RNA is about to go out in the cytoplasm, and in the cytoplasm we have some pretty aggressive enzymes out there that can harm the messenger RNA. And so to protect that messenger RNA, we add a cap and a tail. Once a cap and a tail has been added and introns have been removed, the messenger RNA is now ready to leave the nucleus and go out to the cytoplasm to start translation. So here is your messenger RNA, and if we remember, we said that the messenger RNA, we read it 3 bases at a time, which we call codons. Once you have your messenger RNA, it goes out in the cytoplasm where it's going to meet up with the organelle known as the ribosome, and it's going to start getting translated to make the protein. To make a protein, proteins are made out of amino acids. So as we said before, each codon is going to code for an amino acid. Those amino acids, they start linking to each other. And as you can see, eventually that chain of amino acids will start twisting and it will start having a 3-dimensional shape, and we eventually call it a protein. How do we figure out the amino acids that would result? You have to look at the messenger RNA and you have to look at the codon chart to figure out the amino acid that would result. So right here inside of the nucleus, we have our long 6-foot long DNA. We are not going to read the whole entire DNA just to make one protein. And so the RNA polymerase will have to find the starting sequence for the gene that you want to transcribe, and that would be the promoter. Once RNA finds the promoter sequence, we are going to go ahead and start building the messenger RNA until we stop at the terminator sequence. Once we have the messenger RNA, it's going to be processed. You're going to add a cap and a tail, remove the introns, and it's going to leave. Once it's out, it's going to meet up with the organelle known as the ribosome, and translation is going to begin. Translation also has 3 steps: initiation, elongation, and termination. We also have other players that are going to help us out with our translation process, a bunch of enzymes, and what's an RNA known as tRNA, or transfer RNA. This is how a transfer RNA looks like. What's the point of having a transfer RNA? The transfer RNA is the one that's going to bring over the amino acid. So the transfer RNA is the one carrying the amino acid. As we said, when we are translating our messenger RNA, each codon on your messenger RNA codes for a specific amino acid. Who's going to be bringing over those amino acids? The transfer RNA. On one side, there's the amino acid on the transfer RNA, and on the other side of the transfer RNA, there is what's known as the anticodon. The anticodon is going to bind to the codon of your messenger RNA to bring the amino acid over. This is how the ribosome looks like, and the messenger RNA comes and binds to the ribosome, as you can see right here. The tRNA comes over to bring the amino acids. And as you can see, several amino acids are going to be brought over by several transfer RNAs, and the amino acids, as you can see, start growing and they start linking to each other. So if we zoom in on the messenger RNA right here, the messenger RNA carries a bunch of codons. Now, the very first step of translation is known as initiation. What is initiation? That's where you have to find your starting codon. So to start translation, we must find what's known as the starting codon on my messenger RNA. The starting codon is AUG. So if this is the messenger RNA right here, and AUG is right there, then you're not going to worry about that part of the messenger RNA, and you're only going to start reading the messenger RNA right there, because that's where you have your starting codon, which is AUG. What's going to bind to your starting codon on your messenger RNA? The transfer RNA. How is it going to bind? Well, it carries an anticodon that's going to bind to the codon of your messenger RNA. Once the tRNA binds, it's going to start bringing over those amino acids. Here is the codon chart. So to start translation, we must find the codon AUG. That's where we start. And actually, AUG codes for an amino acid known as methionine. So you're going to be starting the translation process and bringing over an amino acid known as methionine. Once translation has begun, we are going to go through the second step known as elongation, where we are going to keep building that amino acid chain. Now, we are going to keep building that amino acid chain until we hit a stop codon. We have actually 3 different stop codons. If you hit any of those stop codons, translation comes to a stop. So right here is your messenger RNA. It's bound to the ribosome organelle. As we said, the first step, initiation, started when we found the starting codon on our messenger RNA, and a tRNA carrying methionine came and it was able to bind to that starting codon. Then the second step is elongation, and elongation, as you can see, this is where several tRNAs keep coming over, bringing amino acids, and you keep binding all of the amino acids together, creating a long chain. Once, by the way, a tRNA brings over the amino acid, it just takes off. Okay, so tRNAs, their main job is to just deliver the amino acid for you. That's their job. Bring over the amino acid, then take off. That process and the amino acid chain keeps getting longer and longer until we hit the stop codon. Once we hit the stop codon, that's it. Everything comes to a stop. Ribosomes, they are going to let go of that messenger RNA. The tRNAs are going to take off, and you're going to have your amino acid chain, which is eventually going to start twisting itself, and it's going to become a mature functional protein. So right here, let's say that you have your double-stranded DNA right there. Here are the two complementary DNA strands. Now, which strand are we going to go ahead and read to begin transcription? We must read the template strand. So this is the strand that we are going to read. When you read the template strand, you are going to build a messenger RNA. Here is the messenger RNA that you are going to build by reading the template strand. And again, that process of building a messenger RNA is known as transcription. And we already talked about what's going to happen in transcription. You must find the promoter sequence to start it off, and you're going to build a messenger RNA strand until you hit the terminator sequence. And then the messenger RNA has to get processed. Let's say here is the final messenger RNA after it got processed. To begin translation, you have to find the starting codon. So let's say you have right here some codons before the starting one. Are we going to go ahead and translate those codons? The answer is no. You're not going to worry about those. You're only going to start only if you find AUG. Once you find AUG, or the starting codon on your messenger RNA, that's where translation begins. When you start translating the messenger RNA, as we said, you're going to have to refer back to the codon chart, and you're going to read each codon, and you're going to have to figure out which amino acid is it going to code for. Now, who's going to be carrying the amino acids for you? Your tRNA. So the tRNA, on one end of the tRNA, you have the anticodon, which is going to bind to your codon on your messenger RNA. And then on the other end of your tRNA, it's going to be carrying the amino acids. You're going to keep building your amino acid chain until you stop translation. And to stop translation, you have to hit a stop codon. Once you stop your translation, that's it. Everything comes to a stop. Translation stops. And here is going to be your amino acid chain. Stop is not an amino acid, by the way. Stop is just telling you stop right here. Now, never read the tRNA to build your amino acid chain. Remember, the tRNA is just the delivery, right? the tRNA is just delivering the amino acids. So to figure out the amino acids, you must read the messenger RNA because that's the one carrying the message. And so you find your starting codon, you keep building your amino acid chain until you hit your stop codon.